Hello, everyone. My name is Kuok. I'm a field application scientist from MGI, subsidiary group from BGI Group. So today I am going to talk about our DNB microbial detection platform and explaining a few cases from our sequencing users showing how they use our total solution to enable the microbial identification. So first of all, I would like to thank you, LabRoots, inviting me and our company to give this presentation. So today's webinar is going to be divided into 4 parts. First, I'm going to introduce the BGI and MGI history and our DNBSEQ technology. Second part, we'll go for the MGI microbial solution. And the third part, I will discuss about how our BFI or bacterial fast identification works and the case study from it. And the final part will be another bioinformatics solution, which is a pathogen fast identification system, or PFI. So as you may have heard of BGI, Beijing Genomic Institute, it is established in 1999, representing China to contribute 1% of the Human Genomic Project. After that, we decided to drop the name Beijing Genomic Institute, shortened as BGI Group. Over the last 20 years, we have been making tremendous contribution to different human genome projects. For example, in 2002, we contributed 10% to the International HealthMap Project, and we contributed the first Asian Genome Project, 1,000 Genome Project, and just last year, we published a Chinese population scale genomic study with 140,000 genome. So the key point or the key technology of BGI Group rely on the people and the technology, which is coming from MGI. In 2013, BGI Group acquired Complete Genomics, and 3 years after that, MGI is established. So this year is 2019, and you can tell within only 3 years, we have been growing rapidly. For now, we have over 900 employees across the world. Over 40% are R&D people, and there are several hundred patents in MGI as well. So far, we have more than 300 users and more than 1,100 sequencers. So you may wonder why the sequencing user choose our sequencer, because we offer a different sequencing technology. So the sequencing technology from MGI is called DNBSEQ. So DNBSEQ stands for DNA nanoball sequencing. The core of this DNBSEQ technology is actually Warring Circle Amplification, or RCA for short. So what it means for RCA is that in a regular cases, you make a sequencing library, say you start from DNA or RNA sample, and then you make it end repair and A tailing. You add the adapter, and you may do PCR or you don't do PCR, and then some further follow up with some purification, and then you're good to go with a double-strand linear form library. In our case, which is quite different, we denature the double-strand library that you made into single strand and circularize it. So as you can see here, we have the single strand circularized DNA circle. With this circle, we use 529 polymerase to replicate many copies from the original template. So as you can see here, 1 copy, 2 copy, all the way until 300 to 500 copy. So all this copy is coming from the original template. And so this one single strand circle become fat-free DNA nanoball, or DNB, in the Eppendorf tube. So you may wonder why we needed to take an extra step to make DNB, or single strand DNA library. This is because, try to imagine if you only have one circle, it's more like one match, and then you have the weak or low intensity from the sequencer. And in our situation, we make RCA, we make a DNA nanoball format. So it becomes 300 to 500 copy. So it's more like a bonfire. That's why the signal or the intensity is much higher. And then you may wonder, we could have just done it with PCR, just like any other sequencer in the market been doing. But there might be problems, as you may know, with PCR steps in the sequencer. For example, it may introduce false SMP, it may introduce false indels, They might have coverage bias as well, or GC bias. And if you're dealing with many samples in one sequencing one, you might heard of the problem of index hopping, like saying the sequencing width was aligned with another sample, which should have come from sample from the first one. With our DNBSEQ technology and the RCA reaction, we offer a different feature from it. So you may know this is a linear amplification, and there will be really low amplification bias. And because of the way that we amplify the original copy, there's no amplification of the chromosomal error. Say if you make like a mistake from one copy, the next copy that you generate will still coming from the original copy. And because of this, we have much lower index hopping rate. So DNA nanoball, or DNB, is just part of the DNBSEQ technology. We also have the pattern array and the CPAS sequencing technology. All together in this DNBSEQ technology, we can achieve higher sequencing accuracy, better or lower decreased duplication rate, and we also have a lower and actually much lower index hopping rate compared to other people. So we bring all this advantage from the DNBSEQ technology to our MGI Microbial total solution. So when we talk about microbial detection, there are two options for it. One on the right-hand side is to isolate and culture the bacteria, followed by 16S sequencing, or using the bacterial whole genome sequencing, or WGS for short. The idea of this is to detect or monitor, trace the source of the disease, and improve the prevention and the control system. And the other option is the metagenomic sequencing. So in fact, there are more than 99% of the microorganisms in nature. They're not be able to culture or purify under the lab condition. And this type of sample, in order to sequence it, you have to use this culture-free meta sample to perform the metagenomic sequence so that we can identify the unknown microbial infection from it. So with MGI, we offer a series of products starting from the original sample, say blood, feces, flow swab, CSF, or others common sample that you know. We could do manually DNA or RNA extraction from it. We can also do automation to automatically extract the RNA or DNA from it. And as I just mentioned, there are two options. You can go for a single bacteria or the metagenomic sample. So on the right-hand side, the single bacteria sample, you could prepare a DNA library using our kit, followed by the sequencing and the bioinformatics solution that we offer. On the left-hand side, the metagenomic sequencing options, there are three possible ways to do it. You could only, say, extract the DNA from it and prepare a library from it, or you could only work on RNA, or you could work on both, so RNA and DNA in combination to actively to detect those actively expressed pathogens. So after building the library, either the DNA or RNA, the next step, as you may know, is go for sequencing. If you have many samples and you want to have a lower cost from it, you might consider our MGISEQ 2000, which is a desktop sequencer. If you have lower throughput and you want to aim for faster detection result, then our MGISEQ 200, which is also a desktop sequencer, may be good for you. So after sequencing, which depends on the read length, may take 1 or 2 days to generate the FASTQ file, you could use this FASTQ file to connect it to our bioinformatics workstation or server. So there are two systems here for the data analysis of bioinformatics solution. One is the bacterial fast identification system, we call it BFI for short. The second one is the pathogen fast identification system, or PFI. So the difference between BFI and PFI is that if you want to look at single bacteria, then you might consider BFI, and you could do your sequencing with the BFI system to detect the serotype, look at the evolutionary traceability, drug-resistant gene identification, and etc. Say if you have a meta sample and this is something that cannot be cultured, you might want to consider our PFI system, pathogen fast identification system. They're good for blood, stool, throat swab, as I mentioned, and other samples that you're welcome to try. And these application is suitable for infection that are caused by unknown pathogen. And I'm going to give some example in the later slide. And so far, the BFI system is really good for research, hospital, CDC, Center of Disease Control, and cross-border. So let's talk about the BFI system. So BFI is a bioinformatics system. After your sequencing one, you plug it into your bioinformatics workstation with only one click to tell, okay, this is the sequencing one that we just did. Please identify the species format. And within a really short time, it'll give you a genome identification report, which look like that. The first section is about the basic information, and the second section will give you the resistant gene from the sequencing one. The next section on the right is the identification of virulence gene. And the fourth part, which is the one I really like, is to tell you the position of your sample in a phylogenetic tree. And the fifth part will give you the analysis of potential outbreak. As an example here, on the left-hand side, the red square here is the disease outbreak that happens in 2007. And on the right-hand side, is another outbreak that happened in 2012. So if you want to make a comparison between next generation sequencing and the traditional method, which could be PFGE pattern, so PFGE is pulse field gel electrophoresis. So here is the comparison. So the upper part is the protocol or steps from PFEG. Let's assume that you start from day 1 at 8 a.m. So first of all, you will make a gel, which indicate here is the red bar you're looking at is the manual preparation step that you need to look at. And the green part is some sort of water bath or let the machine running without hands-on. As you can tell, it goes through many steps from the left to right until day 2 to get the gel running overnight. And then you look at the result, compare the patterns, and you might get the result in the afternoon of day 2, which is around 3:30 or so. And in the case for NGS, you're looking at, let's assume that you also start from day 1 at 8 a.m. You start from preparation of your DNA sample and going through our automation pipeline, the first phase, and then quantification, and the second phase. With that, you can send the sample to sequencing on the same day, and the next day early morning, because you're connected to the bioinformatic workstation as well, and that's why it generates the result automatically. So pretty much on day 2, when you come back for work, you get the result. So which is like 5 a.m. in the morning. I assume that you show up at 8 a.m. So with that, you can tell the time is shorter. It's like several hours shorter, which might be critical for your case. Not just the time is shorter, but also the hands-on time is less. So you can tell the red bar here and in the PFGE situation, and the red bar here in the NGS situation. Also, not just the time and the room for less error, also is about high throughput that you could go with NGS, because for PFGE, there are like 10 or 15 routes that you could load, but with NGS system, you could actually put several replicate and many patient sample in one sequencer here with MGISEQ 200. So with all this advantage, is trying to outcompete the traditional method with our MGI Microbial Total Solution. As a real case using BFI system, so this is a case that's not published. A hospital in China use our BFI system, use our MGI Microbial Total Solution with the BFI system. First of all, they culture their Microbacterium tuberculosis sample on the first day, the orange part here in the circle. And the next day, the same day, they prepare the DNA library format, and then going through the sequencer, which takes 48 hours in this case, followed by the bioinformatics solution. And in the end, they were able to identify the MTB complex from other microbacteria from the clinical sample. Not just that, they can actually detect 15 drug-resistant gene here from the table of 8 kind of drug. And these tables actually really help them to guide the clinical tuberculosis therapy. So the second case is about a public health issue here, which is published in 2017. So gentleman returning from Angola to China showing some symptoms related to Rift Valley fever. And the hospital decided to do a whole genome sequencing for me to identify the RVFV isolation. So what they do is they going through our Microbial total solution platform followed by BFI. So what it does exactly is to taking the sequencing raw data, which is depicted on the right-hand side here, running through the bioinformatic pipeline by itself to classify the virus, to identify the subtype, and to assemble the genome. And they also look at a whole bunch of things that will be reported in the report, which include evolutionary analysis, size polymorphism distribution, and key size analysis. So the result is that they actually have like 100% alignment of this RVFV isolate. They call it RVFV isolate from Beijing because this is where it comes from. They have 100% identity of 3 gene segment and 98% homologous to RVFV isolate in South Africa. So as you can see, our Microbial total solution really help this public health issue to monitor or actually prevent the disease outbreak. Now we move on to discuss the PFI system or the pathogen fast identification system. So PFI system is another bioinformatics software for our total solution platform. So what it does is to run through the pipeline and then generate a report to help you to identify the microbial genome sequence from the original sample. It's a fully automated workflow. It generates a report within 6 hours once you hit start. It includes a huge database. It also gives you a comprehensive analysis whether this data is coming from RNA or DNA, single end or pair end. So this is how PFI works. After sequencing raw data, it starts to remove the low quality data or the adapted sequence from the original sequencer. It also removes the host data because if you've done metagenomic sequencing, there are high percentage of the sample could come from the host, which could be human or animal. We have the reference genome from the database so that it can remove the host genome, whether it's coming from human or animal. So after the removal of those host data, the leftover data will be going through the database for analysis. So in the database, there are close to 20,000 microbial species that they need to analyze in the software. And after doing all this alignment and analysis, it will generate a report that contains QC result, also a summary of the classification of RNA and DNA data, and the identification from it. So this is not something early or premature. Actually, there have been 3,400 clinical samples have been connected and tested with our sequencing platform and the total solution. As you can see here on the right-hand side is the distribution or percentage of the nucleotide. Over 70%, 75% of the sample come from DNA, and the remaining 25% or so come from the RNA sample. And there, on the left-hand side is the pie chart of the sample and disease. There are over 13 types of samples that our sequencing users have tried. For example, plasma, supernatant, bacterial strain, sputum, whole blood, etc. And in terms of disease, they were able to detect a series of disease from all these samples as well. For example, the most common one will be influenza, salmonella infection, and with the system, they will also be able to detect, say, AIDS or Zika virus infection. So this is another sample coming from CDC, Center of Disease Control from China. There was a day that CDC received a 2 sample from a patient that potentially infected when they crossed border. This particular patient or passenger was suffering from diarrhea. So the CDC decided to do metagenomic sequencing with the sample. So what they do exactly is to collect the sample on the first day, which is at Lunaso at 12:40. They start to do DNA extraction and library construction with our automation system the same day, followed by the sequencing one the same day. And then the next day, day 2 at 2:30 p.m. In the afternoon, they were able to identify the pathogen. So on the bottom left side is the pie chart and a snapshot from the PFI report. Over 44%, so 44.3% of the sequencing one come from E. coli. In fact, at some point that the CDC cannot identify the obvious pathogen on the species level. So they keep searching our PFI database. They searched the variance gene database, and they were able to identify a thermostable endotoxin gene in those E. coli. And that's why they inferred it. This particular patient was actually infected by ETEC or enterotoxin E. coli. And this result actually was consistent with that of culture. And case number 4 is about the diagnosis of animal disease. So our PFI system not only can identify the pathogen from human, we can also identify potential pathogen from animal. So this example is not published yet, but basically a group of goats, they were infected with unknown pathogen with a series of symptoms. And our sequencing user decided to take their full swab, followed by our pathogen fast identification total solution. In particular, they look at the DNA and RNA sample at the same time with our automation system, going through the pipeline, sample prep, library prep, sequencing 1, and bioinformatics pipeline. And in the end, a report is generated within 2 days. So here's a snapshot of the report on the right-hand side. The first one, not sure if you can see it, but I pointed out is actually an off virus, which is quite common in the growth. Based on the sequencing read from both DNA and RNA, so 64.2% of the DNA's data mapped it back to this off virus, and 44% of the RNA mapped it back to that virus. And based on the knowledge that they have, they can tell these symptoms that group of grow had are actually matching the symptoms from this off-virus contamination. And of course, the result was further confirmed by PCR later. So this PFI system really helped our user to diagnose the animal disease. The next case, case 5, is about the diagnosis of rare pathogen. So last year on August 20, a gentleman was sent to hospital. He has a series of symptoms, severe headaches, high fever, high level of the CSF leukocyte protein level. And a day after that, it's getting worse, and he was sent to ICU, intensive healthcare, intensive center unit. And they have a CT scan result for him, getting some symptoms from the brain as well. 4 days after, they took the CSF sample from the culture, and the result from the culture suggests it was negative for bacteria and fungi. So it has been almost like 11 days now, and they went through all this traditional method, and they have problem identify the root cause of this infection. And in the end, the hospital decided to use our MGI Microbial total solution. In particular, on August 31st, which is 11 days after that, they took the sample running through the pipeline from MGI, and we were able to generate a report 2 days after that. So here's the report. On the right-hand side, the bottom right, there's a purple pie chart from it. So the purple part, which account for around 92%, the sequencing one coming from the host, which is human. And the small slice of that pie is the non-host data. If you make it bigger, you can tell the green pipe on the right-hand side, and that tiny bit of slice from the green is actually the sequencing with coming from this really rare pathogen. which account for only 0.0266% of the sequencing one from his sample. So after studying of this bacteria or pathogen, hospital staff were able to identify actually this might be the cause of this infection, and that all happened after 13 days. So basically, my point is that they spent 11 days with the traditional method and they failed to detect the pathogen. With the sequencing one that we provide, it only takes 2 days for them and they were able to identify the pathogen. So if they first decided to go for a microbial total solution, within 2 days, they might identify the rare pathogen and also come up with a possible solution with the patient, other than letting him waiting for 11 days without a better treatment. So we will not just be able to identify the rare pathogen. With the system, you can also identify a lawful or variant pathogen strain. So this is another case coming from our sequencing user, and the paper was published 2 years ago. In this case, a 4-year-old boy was sent to the hospital with some symptoms with the hand, foot, and mouth disease, or HFMD. And the routine solution or the routine diagnosis would be QRT-PCR in this case, to look at whether he is infected with EV71 or CVA16. In fact, the QRT-PCR suggests it's actually a pan-HEV infection. So the hospital decided to use our MGI pathogen fast identification system. So in particular, they go for all the sample prep, library prep, automation, sequencing one, and bioinformatics. The results is shown on the right-hand side in the graph here. There were non-overlapping contact mapped back to genome of the 3 pathogen. First one is human enterovirus C96, and the second one is human poliovirus number 1, and the third one is human Coxsackie virus A24. So ultimately, they also went single sequence with that. The result is shown here in green, and they match quite well with the next generation sequencing data, which is shown here in orange. And the PCR results suggest that all the content actually belong to a consensus sequence to a new strain, which is EV96. And this is actually the first time that metagenomic sequence have been used to identify EV96 as a strain that causes hand, foot, and mouth disease, which is again something that not be able to done with even QRT-PCR. And then next case, case number 7, is the identification of co-infection, which is published in 2018. So in this case, a 26-year-old woman was sent to the hospital. She has a series of symptoms, and heartbeat is super high, 144 beats per minute now. They went through a lot of tests. It was negative for bacteria, fungi culture-based tests, G-test, GM tests. They're all negative. And the hospital staff decided to run another series of tests, in this case, ELISA, qPCR, QRT-PCR, to detect HIV, HPV, influenza virus, SARS, MERS, and other viruses, and the result is all negative. So what they decided to do is to use MGI microbial total solution. So they took her pulmonary secretion from the patient, collected on the first day when she was sent to hospital, using this sample to go through the metagenomic sequence to determine the cause here. So as you can tell on the right side, the pie chart, there were actually 2 viruses, human rhinovirus C and the human Broca virus. They were identified as high abundance using sequencing. And these data or this result were further confirmed by a QRT-PCR. So, and that's why a report was generated to diagnose it is an acute co-infection of both virus. So basically from this case, I want to point out that with the traditional method, you went for all these tests when it's negative. The that really showcasing or emphasizing the power of metagenomic sequence. Because in the metagenomic sequence, there's no previous knowledge is required, so you don't need to decide whether you need to do a culture or what condition of the culture, or ELISA or PCR. All you need to do is just to use the culture-free metasample, going through the pipeline, going through sequencing and bioinformatics to identify the possible cause of this infection. So in summary, I presented 7 cases using our BFI or PFI system with our DNBSEQTM microbial detection total solution. So by saying total solution, we have a series of product line from sample prep, library prep, automation, and DNBSEQ, as well as a powerful bioinformatics solution, BFI and PFI. Although there are 20,000 microorganisms in the database, and we know there's always not enough, and that's why we've been constantly working with our sequencing user to update our database for a better outcome. And as far as we know, our sequencing user or the total solution user were pretty happy with the result, not only because they can get the result within a relatively short time compared to traditional method, which take 10 days in some of the cases that I mentioned, but also because with our solution, they were able to identify the microorganism there, such that they can screen for active microorganism. And in a lot of cases from the CDC or hospital, they will actually be able to use our solution to monitor or sometimes prevent the epidemic disease outbreak. So with that, I would like to thank you for watching this webinar. If you have any question, you can send me an email. My email address is at the bottom left corner there. And if you would like, you could also follow our LinkedIn or Twitter page by scanning the QR code here. Thank you so much for listening and watching. Thank you.